Welcome to Sciencera. Today's video will be focused on Introduction to Immunology. So, in today's video, I will be discussing chapter number 1 from Prescott's Microbiology. This is specially for students who are doing BMI2607 modules from UNISA. So the reference to Unit 1, which is Introduction to Immunology, for you will be chapter number 1, page 1 to 16, and chapter number 32 from this book, page number 707 to 718. The reference book I will be using is 11th edition. If you don't have this edition, you can always refer to other books. I highly recommend you guys to read pages 1 to 16 before watching this video. Let's begin. So, starting with the definition of microorganisms. Microorganisms, as we all know, are the organisms that are too small to be seen clearly without a microscope. So, we cannot see microorganism with our eyes if they are unaided. Microorganisms can be acellular or cellular in nature. Cellular microbes are usually smaller than 1 millimeter in diameter, often unicellular, and if multicellular, they lack the differentiated tissues. Some examples of cellular microorganisms or biological entities are fungi, which include yeast and mold, protists, algae, protozoa, slime molds, bacteria includes E. coli bacteria, archaea. Methanogens are the examples of archaea organisms. Acellular organisms include viruses, which are composed of proteins and nucleic acid, viroids, composed of RNA, satellites, composed of nucleic acid enclosed in a protein shell, and prions are composed of proteins. We're going to look at each of these in details in this video. The difference between prokaryotic and eukaryotic cells. We can remember this by the help of DORA. D for DNA, O for organelles, R for reproduction, and A for average size. So, in prokaryotes, DNA is naked, circular, and there is no introns, while in eukaryotes, there is DNA bounded to protein, DNA is linear, and they usually have introns. Looking at organelles now, there is no nucleus and no membrane bounded organelles in prokaryotic cells, while there is nucleus and membrane bounded organelles in eukaryotes. Reproduction occurs using binary fission in prokaryote and using mitosis and meiosis in eukaryotes. Average size in prokaryote is smaller, which is 1 to 5 micrometers, and in eukaryote they are larger compared to prokaryotes, which is 10 to 100 micrometers. So, to learn the difference between or to remember the difference between prokaryotic and eukaryotic cells, you need to remember DORA. If I have to say the difference in using one line, I would be using the word floor plan to discuss the cellular architecture of these cells. So, for prokaryotes, I will say that prokaryotes have open floor plan, which means that the cell is not divided into compartments. The organelles are not membrane bounded, so they have open floor plan. To say eukaryotes, we say that eukaryote have closed floor plan, which means that cell is divided into compartments. The organelles in eukaryotes are membrane bounded. The membrane bounded organelles allow some cellular material and processes in eukaryotic cell to be separated from one another. These observations eventually led to the development of classification scheme that divide organism into five kingdom system. So we said that organisms can be divided into two types of cells, prokaryotes, either they have prokaryote cells or they have eukaryote cells. So we can say that they are prokaryotic organisms or eukaryotic organisms. Prokaryotic are very tiny, simple cells, no true nucleus, no organelles. And eukaryote cells have larger, more complex cells, have nucleus, have numerous cell organelles. They are further divided into organism with cell wall and no cell walls. With cell walls can either be autotrophs or heterotrophs, and with no cell walls, organism can either be unicellular or multicellular. All these observations eventually lead us to the development of classification scheme that divided organism into five kingdoms. So the five kingdoms were, or five kingdoms are Monera, Kingdom Monera, Kingdom Plantae, Kingdom Fungi, Protists, and Animalia, or we can say kingdom with animals. In this scheme, all organisms with prokaryotic cell structure were placed in Kingdom Monera. The five kingdom system was an important development in microbial taxonomy, but it is no longer accepted by microbiologists. This is because prokaryotes are too diverse to be grouped together in a single kingdom. These studies and other led many taxonomists to reject the five kingdom system in favor of one that divides cellular organisms into three domains. So let's look at the three domains of life. Three domains of life are bacteria, archaea, and eukarya. Three domain classification system was based on three things: 16 sRNA sequences, cell membrane lipid structure, and their sensitivity to antibiotic of members of three membrane. Carl Woese discovered that three domains of life diverge from common ancestor, each with distinct conserved unique RNA sequences. Within prokaryote, there exist two distinct group of organisms, bacteria and archaea, and the eukaryotes have the eukarya domain of life. The comparison of ribosomal RNA nucleic acid sequence began by Carl Woese in 1970. This was instrumental in demonstrating that there are two very different groups of organisms with prokaryotic cell architecture, as I said before, which are bacteria and archaea, and the other one is eukarya. The first domain of life is bacteria. Some characteristics of bacteria are they are prokaryotic without nucleus. They have peptidoglycan cell wall, cell membrane, ester-linked lipid with D-glycerol. They have straight chain, 70S ribosomes, introns and histones with basic protein associated with DNA packagings are absent, and they are sensitive to common antibiotic. To summarize this, we say that membranes of domain bacteria are usually single-celled organisms. Most have cell wall that contain the structural molecule peptidoglycan. Bacteria are abundant in soil, water, and air, including sites that have extreme temperature, pH, and salinity. Bacteria are also major inhabitant of our body, forming the human microbiome. Next one is archaea. Member of domain archaea are distinguished from bacteria by many features, most notably their distinct rRNA sequence, lack of peptidoglycan in their cell walls, and unique membrane lipid. Some have unusual metabolic characteristics, such as ability to generate methane gas. Many archaea are found in extreme environments, including those with high temperature and high concentration of salts. Although some archaea are member of community of microbes involved in gum disease in human, their role in causing disease has not been clearly established. So, we can summarize archaea. They are prokaryotic. Peptidoglycan cell wall is absent. Cell wall is made up of different type of polysaccharides and proteins, and some cell membranes ether are linked to lipid with L-glycerols. They have 80S ribosomes, some introns and histones are present, and they are not sensitive to antibiotic, that is sensitive to domain bacteria and eukarya. Next one is domain eukarya. They have true nucleus, cell wall cellulosic in plants and chitinosis in fungus, and their cell wall is absent in plant. Cell membrane ester linked lipids with proteins. They have 80S ribosomes. Introns and histones are present, not sensitive to antibiotics or has a distinct antibiotic sensitivity profile. Domain eukarya includes plants, animals, and microorganisms classified as protists or fungi. Protists are generally unicellular but larger than most bacteria and archaea. They have traditionally been divided into protozoa and algae. Major type of protists are algae, protozoa, slime molds, and water molds. Algae are photosynthetic. They, together with cyanobacteria, produce 50% of plants' oxygen and are foundation of aquatic food chains. Protozoa are usually motile, and many free-living protozoa function as the principal hunters and gazers of the microbial world. Slime molds are protists that behave like protozoa in one stage of their life cycle but like fungi in another. In protozoan phase, they hunt or engulf food particles, consuming decaying vegetation and other microbes. Water mold or are protists that grow on the surface of fresh water and moist soil. Next, we have acellular agents. Viruses are acellular entities that must invade a host cell to multiply. The simplest virus particle, also called virions, are composed only of proteins and nucleic acid and can be extremely small. The smallest is 10,000 times smaller than a type of bacterium. However, their small size belies their power. They cause many animals and plant disease and have caused epidemics that have shaped human history. Viral disease include smallpox, rabies, influenza, AIDS, the common cold, and some cancers. Viruses also play important role in aquatic environments where they play a critical role in shaping aquatic microbial communities. Viroids are infectious agents composed only of ribonucleic acid. They cause numerous plant diseases. Satellites are composed of a nucleic acid enclosed in a protein shell. They must co-infect a host cell with virus called a helper virus to complete their life cycle. And prions are infectious agents composed only of proteins and are responsible for causing a variety of spongiform encephalopathies such as scabies and mad cow disease. So these are all microbial acellular infectious agents. Next, we're going to summarize what we learned today. Members of microbial world. Point number A we said is microbiology is study of organism too small to be clearly seen by unaided eye. These include viruses, bacteria, archaea, protozoa, algae and fungi. Some microbes, for example algae and fungi, are large enough to be visible but are still included in field of microbiology. While these may be multicellular organisms, differentiated tissue are absent. Then we said that prokaryotes have a relatively simple morphology and lack a true membrane delimited nucleus, while eukaryotes are morphologically complex and have a true membrane enclosed nucleus. Point number 5 was that the work of Carl Woese and his collaborators suggested that organisms fall into one of the three domains based on nucleotide sequence similarities among small subunit ribosomal RNA, which are eukarya, bacteria, and archaea. Eukarya contain all eukaryotic organisms and have a complex membrane delimited organelle structure. Bacteria are diverse and widespread prokaryote which contains cell wall called peptidoglycan. Archaea prokaryotes with cell wall that lack peptidoglycan and have unique membrane lipids. The last point was viroids, virusoids and prions are not composed of cells and are not part of the domain classification scheme. So we can call them acellular infectious agent. This was all about introduction to members of microbial world. In next lesson, we're going to discuss microbial evolution. Thank you for watching. If you like my video, don't forget to subscribe for more.